Our final speaker for this first, and for me, morning session of our workshop today is Che-Wei Xu. So Che-Wei is currently a postdoc in Philip Benfey's lab at Duke. And prior to joining us at Duke, he was a graduate student with Uwe Oehler at the Max Delbruck Center in Berlin. So today, Che-Wei is going to round out our session by talking about automation for data processing, data pre-processing, and some of the tools that he has developed to do that. So thank you so much, Che-Wei.
Okay. Can everybody see my screen?
Yes, looks good.
Okay, so hello everybody, I'm Che-Wei and thanks for inviting me. And today I would like to introduce a tool that can convert single-cell RNA-seq data, raw single-cell RNA-seq data into ready-to-use quality filtered gene-by-cell or cell-by-gene count matrices. And this tool is called COPILOT. And it features non-arbitrary distribution-based background noise detection and removal. And I will explain like why I think it is important and attractive to have a non-arbitrary like quality control scheme for your single-cell RNA-seq data. So I would like to start today's talk with a little bit background introductions. So my main research focus is in bioinformatics for the single-cell data. And over the past few years, I have constructed this beautiful Arabidopsis single-cell atlas with comprehensive annotations together with Rachel Shahan, our moderator here today. And likewise, we have built the rice root single-cell atlas with high-quality annotations with Dr. Mingyuan Zhu. So given these well-annotated atlases as resources and references, we were able to profile the time course data in single-cell resolutions to track and monitor like plant cells, like plant hormone response in root, and which means that we were able to identify exactly which cell type, which developmental stage are demand sites for the hormone response. And also, given this large amount of single-cell transcriptomes with associated labels and annotations, I was able to train a deep learning model to simulate the entire developmental process of the root. And this deep learning model is able to infer gene regulatory networks that bridge like each transition of states in the system. And so from the regulatory network, we can identify key regulatory elements or transcription factors that explain, that is the key to the transitions, specific transition of states. So as you can see, all these, like behind all these projects, you need to first have single cell data, and you want to make sure that you have high quality single cell data. So that means that you have to generate your single cell data properly, and you also need to process your single cell data properly. And one of the very first steps during the single cell analysis is quality control, like how you filter your cells after you got your after you have mapped all your reads to your genome and have the gene by cell count matrix. Like how do you filter out the low quality cells? So back at the time when I got my first single cell raw data, I was not satisfied with the available tools like the Cell Ranger from 10x Genomics, because it was quite like non-transparent to me as to how they filter the cells. Like what's the rationale behind whether a cell is filtered or is kept in your data set. So, and I was not very confident of like moving forward, like with worries on my mind, like wondering whether if my data is under filter, which means that a lot of noise is still in my data set, or the opposite, it's over filtered, means that we've thrown away a lot of informations, and that's basically a waste of money, time, and possibly we are missing out like potential biological discovery. So I don't want to like deal with that like in a downstream analysis. So I want to make sure that I do the correct pre-processing at the very beginning. So that's the motivation behind that. I want to develop my own like quality filter scheme, and that turns out to be COPILOT. And I apply COPILOT on the Arabidopsis root single cell atlas, the root single cell data, and also for rice. And until today, it has also been tried out on not just the plant cell data, but also in human cancer cell line, and also some non-model organisms like corals and nematodes. And in all these projects, COPILOT has delivered like comparable results as to the Cell Ranger. So I would like to first talk about like how Cell Ranger filter out the cells. So they actually empty drops, which is mentioned in Elena's talk, is actually incorporated into Cell Ranger. So the logic behind the filtering logic behind empty drop is fairly simple. First, they seek out to first define the profiles for the ambient RNAs, and then they try to identify barcodes that deviate significantly from these ambient RNA profiles. And those barcodes that deviate significantly will be considered non-empty cells, and which is the valley cells. But what is really, I think it's problems to me is how they first define the profiles of ambient RNAs at the very beginning. So if you read their papers, you will find a line saying that, oh, we use a UMI count thresholds of 100 to define these ambient RNA profiles. And this number is very arbitrary. And so this is a big red flag for me, because it will subject to changes in how deep you sequence your single cell profile, how you generate your single cell data, and what technology, what platforms are you using. So this is like make me wonder if there's a better options to do quality filtering. And after you run Cell Ranger, you take your data, your filtered count metrics to one of these most famous single cell analysis platform, either it's ScanPy in Python or Seurat in R. You will be surprised that they actually will recommend you to apply another layer of arbitrary thresholds to your data set. So that makes me very skeptical about the entire process. I have no idea like whether my data is over filtered or under filtered. So here are some clear drawbacks of applying arbitrary thresholds. First, you lost information. You don't know whether your threshold is set too low or too high. If it's too low, then you basically introduce noise in your downstream like data. If it's too high, then you basically throw out useful information. And there's also issues about generalization. So if you are just like doing one samples or two samples and applying arbitrary thresholds, doesn't matter. But if you are like Elena, they try to incorporate like data sets from different studies and generated by different labs, and it will be a very diverse data. There will be batch effects. So how do you use arbitrary thresholds on all of them and expect that there will be no issues of like under filter or over filtering? And third, it will be difficult to reproduce, even though it's the same piece of like raw data. People all around the world will use different arbitrary thresholds where they think fit. Then you'll basically have like very different outcomes. So that makes me wonder like if there's an alternative to do this. So my motivation is that why can we like define a threshold based on some biological relevant signals, like signals for dying cells? Like people usually use the elevated mitochondrial expression level to represent dying cells. And why can we rely on that signal at the very first place to filter our low quality cells? So with that in mind, so before I jump into explaining like what's the algorithm and steps of COPILOT, I would like to illustrate like what's the potential like negative outcomes of applying the arbitrary thresholds. So let's say you have 5 replicates. These are 5 replicates of Arabidopsis root data. And then these are the histograms of Unicount distributions. And here we have the annotations. The navy blue is the high quality cells, the gray ones are low quality cells, and the red ones are the dying cells or debris. That is defined by elevated mitochondrial expression level. So as you can see here, if we apply the arbitrary thresholds on all our replicates, then from replicate 1 to 3, you are basically doing overfiltering. The high quality cells are thrown away. And in replicate 4, you are doing underfiltering. So some large chunk of low quality cells is actually maintained in your resulting data sets. And only replicate 5 looks fine. Like it has a reasonable separations between the low quality and the high quality cell populations. So that is a problem of applying arbitrary thresholds on your replicates. You will potentially introduce this kind of bias into your data set because you will try to integrate all these replicates like after you do filtering. And for replicate 4, you will see that it contributes a lot of noise in your downstream data set. So that's something that we want to avoid. And in Benfy Lab, today we have generated more than like 200 single cell data, I believe, and most of them are processed by me. And I have seen like drastic batch effects. So the UMMI distributions like can be really different. And so sometimes it's because like different people generated deep data or something went wrong during the data preparation. I don't know, but I just want to point out that it can be very different in terms of the human count distribution. So if you are still thinking that applying arbitrary thresholds on these drastic batch effect is viable, then you are simply inviting like problems in the downstream process. So that's why I come up with an alternative, which is a non-arbitrary filtering scheme. So the very first step in COPILOT is to first identify group of quality cells in your data sets that you have like confidence in. Like it is represented by maybe it has enriched expressions in markers or genes that usually appear in dying cells or cell debris. And then once you are confident that these bunch of group is dying cells, then you rely on it to draw an initial cutoff. And this initial cutoff will separate your cells into two populations, the temporary high and low quality cells. And then you create reference transcriptomic profiles for both the high quality and low quality cells respectively. And what you need to do next is you look into the high quality cell profile and compare each high quality cell to the two transcriptomic profiles. You basically compare the similarity or correlation. And if you find that the high quality cells is more similar to the low quality cell profile than the high quality cell profile, then that cell will be relabeled as low quality cells. And then after that, you update the reference profile because you've done relabeling. And you basically you iterate the entire process until there's no high quality cells that is more similar to a low quality cell profile than a high quality cell profile. So as you can see, the entire step, the algorithms and ideas behind COPILOT is to let the data speak up for itself. So first, you provide your domain knowledge. We know like we are sure that these bunch of cells are low quality cell dying cells, and this is cells that we want to remove. And then you let the data speak up for itself. So it's very similar to a typical Bayesian process where you provide prior, which is your domain knowledge, to your Bayesian process. And then the Bayesian process will give you estimates or likelihoods. So in terms of COPILOT, it will be translated into an organic boundary between into the low quality cell profile and the high quality cell profile. So as you can see, although there's a lot of like batch effects, like different sample is different, but based on the same rationale, which is the dying cell signal, like it can be applied to all kinds of diverse conditions with very different like batch effects. So how does it compare to applying the arbitrary thresholds? Well, as you can see in sample 4, this is actually, if you apply arbitrary threshold in sample 4, this is actually what you will get from Cell Ranger. So Cell Ranger will only think that so there are two peaks of low quality cell populations, right? So the one on the left-hand side will be recognized by Cell Ranger as cells with like ambient RA, barcodes with ambient RA, but Cell Ranger is not able to detect the low quality cells that is on the right. And you may wondering like, how do I know that Cell Ranger is wrong? Because if you consider the right-hand side, like low quality cell peak is high quality cells, then you will get way too many cells annotated as high quality than you initially loaded into the machine. So that is clearly a mistake. So for sample 5, it is also where Cell Ranger or some arbitrary like thresholds suffer. It is in a case where you have the quality, the distribution of low quality cells and high quality cells, they don't separate as well as the other samples. So in this case, you will tend to get a lot of noise in your downstream analysis if you are not trying out like COPILOT. So I have incorporate COPILOT into a protocol that has two components. The first component is like the read alignment, which relies on Callisto and bus tools, which is introduced in Elena's talk. And then the second component is COPILOT. So the entire pipeline can convert the raw sequencing reads all the way to the quality filter ready to use cell by feature or feature by cell matrices. And it also supports Docker and Singularity for the reproducibility, as stressed in Ben's talk, that it's very important to have these environment or images, Docker images. You don't need to worry about like building up your own computational environment. You can just take the images and then the tool and the pipeline is ready to use. And also, it's very computational efficient because we applied Callisto and Boost tools to align our reads. And so basically, Callisto and Boost tool use pseudo alignment that basically exchange precision or accuracy for the speed. So here we are more we value computational efficiency more because I'm usually dealing with a lot of data, so I will like it to run faster. But if you are more if you want it to be more precise, you can try it out like Star or other tools. And here I want to mention that COPILOT can be applied also to the row count matrices produced by other aligner. So it's not necessary to be Callisto bus tools. And finally, it also provides a summary file, which is comparable to Cell Ranger's output, where you have a lot of stats for cells and sequencing. And here I want to demonstrate one of the results, the comparison between COPILOT and Cell Ranger in this non-model organism. So here is the barcode rank plot of COPILOT. As you can see, the boundaries between different categories, like the high quality and low quality cells, are organic. And in Cell Ranger, it's a hard cutoff. So this is just to remind you that Cell Ranger, they rely on the arbitrary thresholds. And you need to think about whether that is what you want as a biologist, whether that will affect your downstream interpretations of your data. And finally, I want to summarize some advantages of COPILOT over Cell Ranger. And so one of the biggest feature in COPILOT is that user can identify and control the filtrations of low quality cells based on some biological signals. So this biological signal is defined by users. You can use mitochondrial expression level, or you can use genes that is associated with noise or dying cells or cell debris. You can use whatever genes that you think can represent low quality cells. And it also offers options for calling and exporting spliced and non-spliced strips for the calculations of RNA velocity. This is contributed by the Callisto bus tool functions. And so, as you know, RNA velocity is now a hot topic, and it is useful for calculating the pseudo time that suggests the developmental potential, the direction of developmental potential in your downstream analysis. And finally, it's very easy to create like customized genomes and annotation files with my pipeline when compared to Cell Ranger. So there's some cases where in your experiments you will add some reporters like GFP, RFP, and you will like them to be captured in your single cell data. But for that, you need to add the sequence of the GFP and RFP to your genome file and annotation files. And so you need to create a customized genome. And also, in some cases, you want to estimate whether your data set is contaminated by other sources. So in that case, you will concatenate a genome of your system and also a genome of your like potential contaminants. So you will need to concatenate 2 genomes or 3 genomes or more. And in that case, you also need to make a customized genomes. And in this pipeline, it makes it very easy to do that when compared to the one required by Cell Ranger. So after quality control, there's like several steps that you still need to do, which is also qualified, also be categorized as preprocessing. So after quality control, you need to do like normalizations. This is just to correct for the sequencing that's effect. So make sure that the technical variation is removed and only the biological heterogeneity is retained in your data set. And after normalization, you do a batch correction. It also depends on how many replicates is in your like experimental settings, and what is the goal of your biological questions. So it's optional. You may need to do batch corrections or you may not. And then you do dimensionality reductions to summarize your data or help with the visualizations of the data. And there are many tools out there that can help you do each of these steps. So if you're interested in the codes, like how to do it by yourself, I have put all my like Jupyter notebooks, like the codes on the GitHub repository under the name of COPILOT. And so I think that's all. So I think I will wish you fruitful single cell projects in the future. Thank you very much.
Thank you so much, Che-Wei.
Yep.
Can you hear me okay?
Yes, I can.
Okay. I think my AirPods have died. So we have some great questions in the chat here. So first, to kick things off, is something that a few people have asked, and that is, what about single nuclei RNA-seq? Is COPILOT still applicable to single nuclei data?
Yes.
Have you tried it?
Yes, so Trevor, another postdoc in our lab, has tried, or Isaiah, I don't remember, has tried a lot of like single cell nuclei data, generate single cell nuclei data for Arabidopsis root, and COPILOT can handle it just as single cell, chloroplast. Yeah, it's just the same. It doesn't. Yeah, it works.
Thank you. Another question that we've had in asked in different ways from a few people is, what about plant genomes that don't have mitochondrial genes identified as part of their reference? How would you handle species like that? So that might be related to what you were talking about with applications to different data sets. And then the second part of this particular question I'm looking at is, how many genes, if you're not thinking about mitochondrial genes, how many genes might you need to really have a good representation of low quality data? So how much information do you need to have?
It can just be single gene. If you are very confident about the gene or marker, and it has a reasonable amount of like expression level in your data set, one gene is enough. And in the case that you simply cannot find any marker or genes to represent your low quality cells. You can also use arbitrary thresholds like in empty drop, like 100. And it's optional in COPILOT tools that there's a parameter for you to feed that to decide that arbitrary thresholds. So you can first look at the distributions of the UMI counts, and then figure out where's the median like UMI counts for the low quality cell profile, and then you can just use that value. So that is also one of the advantage of these COPILOT tool is that you have options to choose whether you want to use biological signals, or you just want to use arbitrary thresholds. But as far as I know, that CellRanger didn't like offer that options for you. Like as I said, it's quite, because it's commercial tools, it's quite non-transparent to the researchers. So sometimes you will feel very hard to use. Yep.
Okay, thank you. Next question is, could you talk a little bit more about the benefits of pre-processing with COPILOT and how you see those benefits downstream, for example, with clustering? So what
Yes.
do you have? Can you talk a little bit about what happens if you try to cluster using or not using COPILOT?
Yes. So actually, when Bert during the first session was talking about, like they found that there are unknown cells like right in the middle of the UMAP. Actually, we have also seen that, but most of the time it's seen in the data set that is processed by CellRanger. But if you process with COPILOT, the same piece of the same data set you process with COPILOT, that cluster in the middle of the UMAP is gone. Like that is what we realized, that so it suggests that maybe CellRanger didn't filter out those cells, but in COPILOT it was able to pick up those cells that is, you know, that is actually low quality based on the signal, the biological signal you provide. And oftentimes the cells appear right in the middle of the UMAP has higher mitochondrial expression level and lower UMI counts, total UMI count. So that is also signals suggesting that these cells may be like low quality and actually not the real like valid cells or cell types. Yep.
So the way to test that biologically would be to then presumably, if you wanted to make sure you weren't throwing away an unknown population of cells, for example, is maybe you would go and generate new markers and try to test if that's an unknown population.
Yes. So I would suggest that you try COPILOT first, and then to see if those cluster is gone. If it's gone, then the explanation is that okay, maybe CellRanger didn't do a good job on this. And if it's still there and the population has like reasonable amount of UMI counts and has some clear signals like a differential expressed genes or markers, then you go into the lab and test out to validate that, and then maybe a new discovery.
Thank you. Okay, last question, just real quick here, since we've had a lot of questions asking about nuclei data. So just to clarify, for nuclei data, single nuclei data, would you use the same markers to determine low quality nuclei as you would with protoplast data?
Yes, that is a very good question. So actually, surprisingly, you can still use like mitochondrial like expression level in the single nuclei data. It's actually not that clean. You know, you will assume that there's no mitochondrial genes in your single cell nuclear data, but actually it's not the case. Like you can still find traces of mitochondrial like expression level in your data, and oftentimes that will overlap with the low quality cells distributions. And so, or you can use stress signal, like because during if there's some stress signal out there, you can use the genes that is known to associate with stress during single nuclei generation protocol. That is also the options. Or if none of that work, you can just fall back on using arbitrary thresholds. But first, you need to check out the UMI count distribution to see that if your arbitrary threshold is reasonable.
All right, that's all the time that we have for this morning session. Thank you so much, Che-Wei, and thank you to all 4 of our speakers in this first session.